In this presentation, we will take a look at the challenges facing the reintroduction of trout into their native ranges. We will start off by reviewing a timeline of their speciation and examining the history of fish stocking practices in the United States. From there, we will discuss the problems with non-native introductions and the effects of climate change on hybridization. Then we will look at how conservation efforts are attempting to remedy the situation, as well as see some examples of successful reintroductions. Finally, we will wrap up with some points to be mindful of for the future and some general conclusions. Let's get started. We will begin at the beginning. Around 100 million years ago, the salmonids broke away from the evolutionary tree to form the family that contains all modern genus and species of trout and salmon. Somewhere around 30 to 40 million years ago, the genus Salvelinus broke off to form what we now see as brook trout, lake trout, and Arctic char. Two more splits occurred around 15 million years ago to form the genus Salmo, which contains salmon and brown trout, and the genus Oncorhynchus, which contains rainbows and cutthroats. Even though their modern native ranges were not determined until millions of years later, they began to evolve separately in different areas across the world. The Wisconsin glaciation isolated these species within their topography to establish what we consider to be their native ranges today. We will come back to this later, but for now, it is important to know that rainbow trout occupied the Pacific coastal region, cutthroats occupied the Central Rockies, brook trout settled on the East Coast, and brown trout inhabited Europe and the Baltic regions. This arrangement would stand until the 1800s, when humans began to breed and distribute them across the globe. In the post-Civil War era, westward expansion and a connecting railway brought fishermen from the East Coast to the Rockies and Pacific coastal regions. By 1871, the government had created a U.S. Fish Commission to oversee hatchery development and fish distribution in public waterways. Spencer Baird and Livingston Stone, seen here, began breeding rainbow trout, which were ideal because of their ability to withstand a variety of conditions. By 1887, they had transplanted 3 million rainbows to 33 of the 38 states, as well as Europe, Canada, and Mexico. Their hatchery on the McCloud River in California created the ancestry to almost all of the transplanted rainbows worldwide today. What they started with rainbows, others would follow with other species, and before long, a complete corruption of native ranges would ensue. Rainbows were introduced into brook trout and cutthroat ranges. Cutthroats were introduced into brook and rainbow ranges. Brook trout were introduced into western watersheds that previously held rainbows and cutthroats. Before long, you have fish everywhere where they're not supposed to be. What Baird and Stone were unaware of at the time was that these non-native introductions would increase competition among species trying to occupy the same ecological niche. This would inevitably compromise the survival of native species and cause hybridization in some cases. The three main hybrids that occur due to species overlap are cutbows, tiger trout, and splake. The most important of the three for our purposes is the cutbow. Because rainbows and cutthroats are both from the genus Oncorhynchus, their genetic similarity allows for highly successful hybridization. This is detrimental to native cutthroats because it compromises genetic purity within some endemic populations, and the hybrid progeny can oftentimes outcompete their native parents. The other hybrids are rare in nature and highly unsuccessful at passing on hybrid DNA, but it is possible. However, recent research has revealed a new enemy. Global warming, caused by climate change, has become a significant factor that is threatening the survival of genetically pure species. As you can see from the diagram, stream temperatures are rising and snowmelt is decreasing. In this situation, rainbow reproduction is more efficient than the native cutthroats, causing cutthroat fitness levels to decrease. Also, overlapping ranges are causing increased hybridization in areas that are not geographically isolated. Those species that are protected by geographic isolation face other problems. Increasing natural disasters in the form of fires and flooding events that have been linked to global warming can be very destructive to the habitats of endemic species that are vulnerable to extirpation and extinction. Protecting the genetic purity of some of these endemic species has become a rallying point for conservationists, naturalists, and fishermen. To combat the range corruption brought on by Baird and Stone and their followers, restoration efforts are at an all-time high. The three main components of these efforts are habitat restoration, which involves bank stabilizations and revetments, as well as other in-stream modifications to promote habitat zones and control existing stream flow. Streambed modification, which involves a highly invasive reconstruction of stream pathways and rebuilding of riparian corridors. Many times heavy machinery is used and the entire ecosystem is modified. And finally, poison and reintroduction. This tactic uses an organic compound called rotenone, which is highly toxic to fish and only mildly toxic to humans and other mammals. For this to work, sections of streams or even entire watersheds are electrofished to capture native species. The poison removes all the non-natives after it is applied, and the natives are reintroduced when the rotenone has dissipated. This strategy has been implemented all over the United States with great success. Threatened species of cutthroats and rare endemic subspecies have been reintroduced into their native ranges, and large portions of some eastern watersheds are being cleared of non-natives to promote native brook trout. The success stories are great for native species and show the true power of focused and collaborative conservation efforts. So where do we go from here, and what do we need to look out for in the future? The work of focused conservation efforts have been highly successful to this point. The future of native species reintroduction will need to include increased monitoring programs to detect non-native species and non-geographically isolated waterways. Whenever possible, geographically isolated waterways should be explicitly chosen as reintroduction target sites to ensure the perpetuation of a genetically pure species. This can be tricky, however, as genetic bottlenecking can occur when breeding populations dwindle in size. The founder effects of a genetic bottleneck can limit the genetic diversity of small populations as they grow. Oftentimes this will result in potential inbreeding and the abundant recessive gene expression within the population. Moving forward, controlled breeding programs and decisive distribution of dissimilar individuals will be the most productive long-term breeding strategy to ensure the maximum amount of diversity within the remaining population. As we have discussed earlier in this presentation, the legacy effects of careless stocking practices has threatened the extirpation and potential extinction of genetically pure trout species. In a direct way, increased wildfires can destroy limited habitat zones, and we also see how indirect effects of climate change threaten the survival of these fragile species. Rising global temperatures and alterations to snowmelt patterns have caused detrimental effects to the reproductive habits of cutthroats where rainbows often occupy the same habitat zones. Despite all of these hindrances, widespread conservation efforts have shown to be extremely effective for creating and restoring habitat zones and reintroducing native species. Thank you.